everyone, and welcome back to Simcyp technical webinar. My name is Xian Pan. I'm part of the translational DMPK group at Simcyp. I will host the session today. So today's topic focuses on the new features introduced in Simcyp version 24 on the trial design and optimal dose selection. So before we start the talk, I'd like to kindly request that everyone stay muted during the presentation. Should you have any questions, feel free to raise your hand or post your query in the chat box. We will have a dedicated Q&A segment at the end where we will address them all. We are recording the presentation, which will be available on YouTube shortly. Next slide, please. And it's my pleasure to introduce today's speaker, Dr. Iman Ali Khattab. Iman is a research scientist in translational DMPK group. Since she joined the Simcyp in 2022, she has actively contributed to the development of the new populations and compound files. She's been involved in the oligonucleotide module and also the IVIVE. She has led the expansion of the trial design and optimal dose selection project, which she will talk about today. She earned her PhD from University of Manchester and completed a postdoc at University of Liverpool, and she has several publications in this field. I'm glad to have her with us today. Her talk today will cover the new dose finding tool that assists the user in determining the optimal dosing regimens to meet the specific PK or PD target for the defined population, and she will also show some case examples. Without further ado, I will hand over to Iman. Over to you, Iman.
Hi everyone. Today I'll speak about the trial design and as an integral part of the BBPK modeling along with the drug data and the system data. In the trial design, we define the dosing regimen as well as the demographics of the subjects included in the trial in order to improve the predictability of our model. Usually in Simcyp, we expand the clinical trial design features in order to allow more applications and also allow easier and faster use. Today I'll speak about one of the tools that have been introduced into the simulator in version 24, the dose finding tool. The aim of the dose finding tool is to enable the selection of the optimal dose based on a specific PK or BD target, such as the AUC, Cmax, or BD effect. So once you have developed your drug model and you have a workspace in the simulator and you want to define the dosing regimen, either the amount of the drug administered and or the frequency of administration in a certain population, and you have prior knowledge about the desired PK or BD effect. So you want to achieve a certain Cmax value, a certain AUC, or sometimes a certain BD effect. So knowing this prior PK or BD target, you can use the dose finding tool, which will simulate a series of doses or dosing frequencies, and will give you an idea about this specific parameter or BD target for each dose, and you can visually see which of these doses lies within the predefined target range. It also, because it's a population simulation, it will also give you an idea about the probability of attainment, which is the percentage of subjects achieving the predefined range. Sometimes you can define the probability of attainment by 70%, 80%, whatever you would like to do. I'm trying to present two case studies to show how the tool can be used, but of course the applications can be more than these case studies, and you can think of other scenarios and applications based on the drug that you are working on. The first case study is for Theophylline, which has a narrow therapeutic window of a concentration between 10 and 20 milligram in adults. Of course, higher concentration than this will achieve or cause some adverse effect or toxicities. Lower concentration can lead to treatment failure. In the Simcyp simulator, we have the Theophylline file where the absorption is defined by first order, minimal BBK, and the enzyme kinetics for the elimination, as well as renal clearance. The model has been verified with a number of clinical data as the one shown here. So we will use this model to determine the optimal dose using the dose finding tool. The research question that we want to answer is which dose will give the best probability of attainment for the Cmax without causing toxicity for adults. So let's start with this in the tool, so I can demonstrate this in the Simcyp simulator. So we have here the workspace with Theophylline model or Theophylline substrate model. I have also the population, which is the healthy volunteer population. You can switch to any other population. In the trial design, I specified in the workspace 10 subject, 10 trial with a certain age range, as well as the percentage of females, and the duration of the study is 24 hours. For the dosing, I'm starting with the 5 milligram per kilogram oral dosing. Multiple dosing selection was taken here, but because the simulation is 24 hours and the tau is 24 hours, so I have only one dose administered. So what you want to do in order to start with the dose finding is to save this workspace somewhere in your computer and recognize the file where you saved this workspace in. And then going to the workflow, you will see the icon for the dose finding, the optimal dose selection based on a specific BK or BD target. Once you click this icon, so you have the first screen, you will go through a series of screens. The first one is where you define your input file, your workspace that you want to work on, and the output for the resulting output. So the input file, you will go to the place where you saved your workspace. I saved it here. So I just open it. And then in the output folder, I select the place where I want to save my resulting output. So I will save it in the same folder. If you want to change it, so you can go to another folder on your desktop or on your computer. The file name is the workspace name. Here I have the demo drug 5 milligram and so on. So it's the workspace name, as well as the dose range that you will specify in the next step. So it will be a very unique name to your simulation, but of course you can change the name to whatever you want. So next, you will have the second screen where you set the doses that you want to simulate. We have two options. Firstly, to change the dose amount or the dosing interval. For the dosing amount, we have up to 10 different doses that you can evaluate. The unit here is taken from the workspace that you have previously saved. You cannot change the unit here on the screen. If you want to change it, so you need to change it in the workspace before saving it. So it is in milligram per kilogram, and you can define up to 10 different doses. I have here 5 different doses for the demonstration. So you can run the simulation. The second option, sorry. The second option is the, I can run it again. Something happened because I pre-saved it. The second option is the dosing interval, because I have the multiple dose administration as defined in my workspace. So I can change the dosing to the interval, where I can define different taus. Again, we have 10 different dosing interval to evaluate. But in this study, I want to change the dosing amount only and not the interval. For the IV dosing, IV infusion, sometimes for the infusion administration, we give the initial loading dose, which is IV bolus, followed by IV infusion for a certain period of time. So the dose amount will be applied to the infusion, not the bolus dose. Now the simulation has finished, so I have this green tick. I go to the next screen. The third screen, after defining the doses, so we need to define the BK or the BD target. The BK target can be the average concentration or the Cmin, Cmax, different AUCs option, the first dose AUC or the infinity or the last dose AUC. We have also the cumulative AUC throughout the simulation time and the Tmax. If the workspace has the BD model active, so you will see also some BD parameters like the minimum response, the maximum response, the time for the minimum response and the time for the maximum response, and the AUC for the BD profile. So these are the BK and BD parameters. We have also the profiles if you want to see the profile generated by each dose or dosing regimen. So you can have systemic concentration or BD response profile. For the systemic concentration, you have the plasma or the blood, and for the plasma, you have the total concentration or the unbound concentration. The target range can be specified here in this step. So for our example, if you remember that theophylline concentration was from 20 to or 10 to 20. This is the therapeutic window for theophylline. So I define the minimum maximum. If you have only the maximum concentration, so you can define it and just turn the minimum to the left side, or the minimum and turn the maximum to the left side. So you can define both or one of them. For the units, it's important to let's get this back to 10. It's important to make sure that the units are the correct units for this target range. So for theophylline, it is 10 to 20 milligram per liter, but we have also a list of different units that you can choose from, and the time unit in hours. In case of BD profile simulation or BD parameter, we have also the BD response label that you can type in to change the whatever the BD response that you are working on, like the reduction in the blood pressure, reduction in the blood glucose level, or what is the effect of your drug. For theophylline example, we need the Cmax to make sure that it doesn't go beyond the 20, not to cause toxicity. There is also a scaling factor here, which is nothing but a multiplier to your parameter. So if you know that I don't want the AUC itself, I want the AUC divided by a certain number. This is sometimes important for some antibiotics or antimicrobial drugs, where we divide the AUC by the minimum inhibitory concentration of this drug. So we know the minimum inhibitory concentration, which can be, for example, 10 milligram per liter. So we divide all the AUC value by 10. So we need to multiply by 0.1. And this is clarified here. If you hover over the question mark, you have a detailed explanation of this scaling factor. For the time being, I don't have anything to multiply my Cmax with. The other option here is the additional output graph, which is related to the target attainment, the percentage of subject achieving the target. So you tick on this box if you want it, and by default, 70% target attainment is shown here. If you want 100% of your subjects to achieve the target, so you can change this value to this, or 50%, so it can be changed to any value that you want. I will keep it like 70% as the default for now. Then you click on the output. Okay, I did a lot of clicks. But once you do the click, you have the Excel file here. The Excel file of the output has 3 different sheets. The first sheet will summarize the input. So you have the population name, the number of trials, number of subjects. You have the route of administration for your drugs and the units and the simulated doses that you defined in the dose finding tool. You will have also the target and the ranges that you have specified with its units, and also the probability of target attainment. It's a summary of what we have introduced into the tool. Then the summary sheet, which is the summary of the results. It has 2 graphs and also the dose target attainment summary, which is the mean Cmax calculated for each dose, and also the standard deviation. We have also the probability of target attainment calculated for each dose. Looking at the first graph, you can see the dose against the PK parameter that we have identified. So you have here the 5 different doses. Some of the doses are below the range. So this dashed line are the 10 to 20 milligram per liter range that we defined. And some of the doses are within the range. Some of them are outside the range. But of course, not only the mean, but we have the variability around the mean. So the percentage of subject within the range is defined in this graph. And you can see that the third dose is the only one or achieving the probability of attainment above the pre-specified 70%. It reaches to 77%. If you are interested into individual values for each subject in each trial, so you will have detailed calculations here in the third sheet, and you will have also a statistical table, not only the mean, but also the median, geometric mean, and other calculations. So this is the output of the dose finding tool. I'll go back to my presentation. So replying to my research question, I have simulated 5 different doses. I defined the Cmax, the range of the therapeutic window from 10 to 20. I got the simulations for the 5 different doses, and I can see that the third dose, which is the 7 milligram per kilogram, achieved the highest probability of target attainment. So it's probably the best dose to use for this population. The second case study, I'm trying to show another kind of parameters or simulations related to the BD effect. So ciprofloxacin is an antibiotic used for treatment of bacterial infection. And we have a BD model for ciprofloxacin using ADAM absorption, full PBPK distribution. And we have also the BD effect determined by logistic growth model. The BD response is measured using what's called log 10 colony forming unit. The log 10 colony forming unit of 6, for example, means that the number of colonies of bacteria formed after the application of this drug in this dose was 10 raised to the power 6. So the higher the number, the larger the amount of the bacteria formed, so the lower the effect of the drug. However, going down or lower the number of log 10 CFU means that the antibiotic is leading to the formation of lower number of bacteria, so the effect of the drug is higher. The PK and the BD model have been validated using different observed data, and this is one of the simulations showing that we are capturing the BD effect quite well with the oral dose of 500 milligram twice daily. So the research question here is which dosage regimen, either increasing the dosing amount or reducing the dosing interval, that will give the better probability of reducing the log 10 CFU below 4. So now with the 500 milligram twice daily, we can see that it never goes below 4. So if I want more effect, I need to increase either the dose or the frequency of administration. So we have two different scenarios using the dose finding tool. One is changing the dose amount from 500 to 1,750, simulating these different doses. The other scenario is using the twice daily dose and also increasing the frequency by shortening the tau to 8, 6, or 4.5. The target here will be the BD response profile. I want to generate BD response profile for each dose similar to this one. The target range should not exceed 4, and the response here is labeled with log 10 CFU. So the log 10 CFU, I don't want it to exceed 4 as per the research question. Again, the target attainment was set to 70%. These are the results from the dose finding tool. The first or the top graphs represent increasing the dose amount. So from 500 milligram, which is the top profile, going down, we are increasing the effect, we are increasing the dose, and you can see that the highest dose is barely going down below the 4 log 10 CFU. And the probability of target attainment is reaching to the maximum is 40%. The second scenario is keeping the dosing amount the same, 500 milligram, and increasing the dosing frequency or reducing the dosing interval. So the twice daily dose is shown here in yellow, and then going down with decreasing the tau, you can see that the 4.5 hours has reached the levels below the log 10 CFU of 4. And this tau is showing the highest probability of target attainment. When we select the profile, either the BK or BD profile, so now the probability of attainment is calculated not as a percentage of subject achieving the target, but it is the percent of time through which the profile is within the required target. So throughout the simulation time, how long the profile was kept within the range. So from this exercise, we can conclude that reducing the dosing interval will give the better effect for reduction of the forming bacteria using ciprofloxacin. I'm now done with the demonstration of the dose finding tool and happy to receive any question.
Okay, thank you, Iman. Now let's move on to the Q&A session. So we'd love to hear what you're thinking or any questions you may have. You could raise your hand if you'd like to speak, or you could type in your question in the chat box. We will read aloud. So while we're waiting for the questions to come through, I could start with someone. So what's the difference between this dose finding tool and the sensitivity analysis we could do on those?
Yeah, that's a very good question. In the simulator, the sensitivity analysis is using the PopRep mode, which is only the one of the subjects that had been simulated, not as a population simulation. However, in the dose finding tool, we have the simulation for the whole population, so we can have the variability along with the mean. But with the sensitivity, we have only the mean value. So to get the probability of attainment, something like this, or understand the outcome in the whole population with multiple subjects, the dose finding tool will help in this over the regular sensitivity analysis.
Okay, thank you, Iman. We have got a question from Devon asking if the PD model is in Lua, can the dose finding tool be used?
Yeah, that's a good question. As we have this BD model working for the Simcyp simulator, and you can simulate it with the normal virtual population simulation, so it will work with the dose finding tool as well. Yeah,
Okay.
either with Lua or with any other kind of BD model definition.
Okay, I think that. Okay. So maybe just to follow on that, so we were talking about the compound side of the PD module. So what if we made any change in the population? Can those changes be applied to the dose finding tool?
Yes, of course. Whatever you have in the population, whatever you choose as a population or make a change in the system data, it will be reflected to your default population in the dose finding tool. So it uses the population that you introduce here in the workspace that you are working on, and all the system data that you introduce in this population will be reflected in the dose finding tool.
Okay, thank you. I know the talk is really about the dose finding tool, which is the big one. Are there any other new features in the trial design that might be useful to our audience you want to introduce? You want to talk about in the version 20?
Yeah, we have developed different tools related to the trial design. One of them is the use of custom trial design for multiple population. Previously, we had this custom trial design specific for the virtual population simulation. However, in version 24, we allowed the possibility to introduce the custom trial design template to each subpopulation within the multiple population simulation mode, and this will help you get understanding of the changes in the exposure across the multiple populations, and also to identify the subjects included in the simulation individually if you have this data already in the clinical studies, so you can define it more accurately now with the custom trial design for each subpopulation. Another feature is you can use the multiple population mode. Previously, you had the option to set only one dose across the 3 or 4 populations that you are simulating simultaneously. But in version 24, we had added the option that you can vary the dose across the different populations. So you can set a certain dose in healthy and then change the dose in the other population or organ impairment, and decide if this reduction in the dose or increase in the dose resulted in an exposure similar to the healthy. So this is another feature that we have added. And multiple other features like the weight range identified also for the virtual population simulation.
Okay, it seems you get more questions coming through while you introduce all these new features. One question is it possible to select the AUC or Cmax ratio as the PK target for DDI simulations? I think it's asking about the dose selecting tool.
No, at the moment with the dose finding tool, we have only the AUC Cmax. But if you have enabled the DDI, if you have enabled the inhibitor in your workspace, so what you will get as an AUC is the AUC of the interaction. So you can have 2 workspaces, one with the interaction, one without the interaction, and you can try the dose finding tool. It will be just 2 different workspaces, and you can try the dose finding tool once with the DDI and once without the DDI, and the AUC can be AUC ratio can be calculated, of course.
Okay, just want to add to that, there's some new feature in version 25, where you could run the population sensitivity analysis, and where you could actually to look at AUC and the Cmax ratio, and and have more sensitivity on the dose. So hopefully that will be another one you could use. Another question is really a clarification. It's about the dosing finding, whether we could change both the loading dose and the maintenance dose, or just the maintenance dose.
If you have the loading dose and the IV infusion maintenance dose, so the dose finding will apply the series of doses on the IV infusion maintenance dose. And the loading dose will be the same as what you have defined in the workspace. So it applies only on the maintenance dose as clarified in the note.
Another question is about the percentage attainment of patients versus of time. Does it depend on the endpoint of PK or PD?
Yeah, it depends on the target that you have selected. So let me go here. So for the parameter, if you have selected a PK or BD parameter, it will be the percentage of subjects attaining the target. But for the profile, either PK or BD profile, so what's calculated as attainment will be the percentage of time through which the profile was within the target range. So it depends on this selection, parameter or a profile.
Okay. And another question is the new population-specific dosing tool able to select adult and pediatric with different age and weight bands?
And you mean this one, specifying different doses for multiple population?
I think so.
I think, yeah. For the multiple population mode in general, you cannot mix adult and pediatrics at the same time for the multiple population simulation. You can have the 4 populations as adults or the 4 populations as pediatrics, and then you can change the dose. But if you want to try the dose in adult and compare it to the pediatrics, so you need to create 2 different workspaces, and you can of course use the workflow that will run multiple workspaces for you automatically without doing it manually.
I hope that's just your question, Kuna. Okay, so I haven't seen any more questions coming through. Okay, I guess you. Okay, so maybe you could help me move to the last slide. So I didn't. Okay, so really thank you, Iman, for the talk about the dose finding tool, and actually more new features will be covered in this talk. Thank you to everyone for joining us and for attention and active engagement. If you have any additional questions later on, please don't hesitate to get in touch with us. So next technical webinar is scheduled on the 10th of June. It's about the concentration-dependent blood and plasma binding in the simulated version 24. So if you're interested in it, please register through the members area. Once again, thank you for being with us today. We look forward to seeing you soon. Wish you all a lovely day and goodbye.